Hello, everyone. This is Dr. Nordquist. This is January 6, 2022, and I came here this morning to film a lecture on the series of why we get sick. And in reviewing some of the files, I found a lecture I'd done Wednesday, March 21st, 2018 for American Academy of Oral Systemic Health. And this was done right after we made the discoveries relating viruses to spirochetes, immune suppression in relationship to Alzheimer's disease and mad cow disease, which is CJD, Creutzfeldt-Jakob disease, and other chronic diseases. And this was a great discovery at that time, and it got published in Dentistry Today, and this was the first lecture after that publication went into Dentistry Today, relating prions, mad cow disease, viruses, diabetes, Alzheimer's, mad cow disease, and chronic disease. So what I'm going to do is I'm going to play that lecture for you, and you can see the development of our understanding how spirochetes, which I've been lecturing on for many years, and how they carry the viruses that seem to be the instigation of the immune suppression, and then the way the immune system reacts inappropriately to viruses now that they have been suppressed by these particular bacteria. So I find it very interesting, and as I listen to my own lecture and reviewing some of the things that I've been teaching for many years and how it all fits together. So I want you to all watch that lecture and see how this whole thing was first put together, and then in the series of lectures I'm doing right now, how we've developed it from there. One thought before I show you the video from the presentation is that the discoveries that were made that allowed me to publish these papers were totally overshadowed by what happened in the last 2 years. So in my opinion, the greatest discoveries in medicine in the 21st century was totally overshadowed by two political events that occurred in these last 2 years. And you can just see the two pictures I have on the screen so we don't get censored.
Good evening, everyone. Welcome to tonight's AOSH webinar. My name is Ashley Height, and presenting tonight's webinar is Dr. William Nordquist, who is a dentist, oral biologist, researcher, writer, and speaker. And he's here to talk to us about the pathogenic mechanisms that connect oral health, diabetes, and Alzheimer's disease, among others. So thank you for joining us tonight, Dr. Nordquist.
Okay. Can you still see the webinar on the side?
Nope, we can't see that. You are good to go. Before we begin, though, I'd like to remind everyone that there will be time to address questions at the end of his presentation. So go ahead and enter any questions that you might have into the panel on the GoToWebinar panel. So with that, I will turn the mic over to you, Dr. Nordquist.
Okay. Good evening, everyone. I'm Dr. Nordquist, and here I am on my boat in San Diego, California, and it's sunny right now, but we're going to have some rain later tomorrow. But this subject that we're going to be dealing with is prions, viruses, their relationship to systemic disease and the implications to dentistry. So there's a lot of material here, and I'm assuming that many of you have some knowledge of this subject. So to get through this whole thing in an hour, I have to move along pretty rapidly. So this is going to be, evidently, you can access this for credit from now on so that you can go back and review it if you want to. So we'll get started. Here we go. This is actually based on a paper that we just published. It was Dentistry Today, and it went out as a news report. You can access that by going to Dentistry Today and just page through their news reports, and you'll find the paper that this lecture is based on. We're going to talk about, first I'm going to give some examples. There's just 3 quick examples of patients with chronic disease and how we mitigated it. And everything that we're going to talk about has to do with the balance of bacteria or the microbial garden, not only from the oral cavity, but throughout the gut, from the mouth to the anus. And then it's microbial alteration and immune suppression that seems to be the primary underlying cause of this problem. And once I'm going to give quite a bit of information on what I think is the underlying microbe that seems to be the instigator and the carrier of the viruses that seem to be doing the alteration, and we're going to talk about how the garden gets altered. Then at the last part, we're going to get into the meat of the paper. It's herpes virus, and then the relationship between Alzheimer's disease, diabetes, and CJD. And then we're going to get into the virus or the spirochetes and viruses and how the spirochetes carry the viruses, and then the prion theory and prion diseases. And so here we go. Here's some patients that have these chronic diseases. If you notice, this patient has alopecia, no hair, and she was referred to me. And so we did our little hygiene thing, and we were able to, within 6 months, she started growing her eyebrows and her eyelashes back. And I'm not taking credit for it because this is an anecdotal, just one case. But it gives you a really good feeling to be able to treat some of these chronic diseases successfully. And it makes your day, believe me, when you see something like this in 6 months. Here's a patient, and she happens to be the husband of one of my editors. And she's a very, very smart person, Bonnie Bennett. And she wrote a book, Tick Bites and MS. And it goes into a lot of the stuff that I'm going to go in. But they called and said, my husband has been going blind over the last few months, and he's totally blind in his right eye. And she thought it was caused by spirochetes that probably coming from the mouth. So I told her, I said, get your butts over here and we'll get a microbial analysis or a microscopic examination and a CAT scan to find out if there's a source of infection in his mouth. And so we did a CBCT, and lo and behold, we found tooth number 3 completely abscessed. I referred her back, or him back, to Tucson, where they had an oral surgeon who went in and cleaned that whole mess up and said it was one of the worst abscesses that he'd ever treated. And also, she put him on doxycycline, which she feels is very important in treating these spirochetes, but long-term doxycycline. And within 6 months, he totally regained the sight in his eye. And here's another patient that came in, and one of my associates had treated him first, and he actually did a root canal on number 3 here with just regular x-rays. And the regular x-rays didn't show anything. There was no sign of sinus involvement whatsoever. It's on a Thursday, and I happened to be in that office on Friday, and she came in, and she basically had Ludwig's angina. She still could breathe, but we got her to the hospital, got her on antibiotics, and they took the swelling down, and then we got a CAT scan, and we could see this huge area of infection, which involved basically 3 teeth, 3, 4, and 3 and 4. We end up, it was 2, 3, and 4. And so we did root canals on 2 and 4 and extracted number 3, and now she's doing really well now. But anyway, what we're going to be talking about is the balance of bacteria or balance of microbes from the oral cavity to the anus. And it's something that's actually gone haywire in our modern society. And we're going to give some answers as actually how this garden got so screwed up, to be honest with you. But anyway, the alteration of the human microbiota, we're going to talk about antibiotics, diet, and human migration. My co-author, Dr. Krutchkov, and I wrote a paper about what's called the metagenome and its relationship to periodontal disease. As Connecticut Dental Journal, and you can go find that if you want to. But it goes into what happens over a lifetime of dealing with microbes, and then microbes that are out of balance. And you accumulate these bacteria or these microbial particles and over a lifetime, and pretty soon it overwhelms the immune system and it depresses the immune system, which leads to these chronic diseases. Probably the biggest clue that I ever had for understanding this balance is this book called Missing Microbes. And I highly recommend you find this book and read it and study it, because Brasler really has insight into what happens when we alter the microbes. And he was dealing with H. pylori, the microbe that causes ulcers and ulcers and stomach cancer. And over a 20-year period, he had the particular microbe. And he did experiments in over a 20-year period and decided, and he and his colleagues decided that H. pylori wasn't so bad. And if we take it away, then you get heartburn. And so he felt that the inflammation that so-called was caused by this microbe was actually a normal reaction in the gut. And the inflammatory cells were actually part of the immune system. And he goes into the long, beautifully written book about how the immune system uses these microbes to modulate the immune attack. And without those inflammatory cells, the body cannot modulate the immune attack according to whatever microbe a patient's exposed to. So the microbial garden, actually 20% of the DNA in the body is actually human. 80% represents bacteria, virus, viral, and fungus DNA. And this garden needs to be tended properly. Things that interfere with it is the modern diet, the effects of antibiotics, and the need to use probiotics. So with that in mind, I began to think, well, what is plaque? If inflammation is good in the gut, maybe plaque is the same function, assisting the immune system to govern or to modulate the immune system in its attack, starting for the oral cavity. For years, I've been looking at high-powered microscopy of plaque, and I have to admit, throughout all those years, I didn't see the trees for the bugs that were running around, and I didn't see the forest for the microbes. And so when you're looking at plaque samples, normal plaque samples, and if you do this over a long period of time, you find that the vast majority of what you see on a microscopic examination of dental plaque are PMNs. They're immune cells. And so I'm thinking to myself, wow, this is something that is normal in the mouth and it's part of the immune system. These PMNs, polymorphic nucleosides, are actually serving the same purpose of sensing what you're exposed to and then modulating the immune system accordingly. So microbial alteration and immune suppression, just to give you an example of what this, how severe this problem is, I get this so simple, I can ask my questions, 3 simple questions. And I ask also health professionals, physicians, nurses, nurse practitioners, and anyone interested in the immune system and how antibiotics affect the body. And I tell them, they're going to ask 3 questions, riddle, and all questions have the same answer. But by the time you get to the second question, most people that know the most about this cannot answer that second question. So the first question, I ask the question, what is the most important discovery in modern medicine that probably resulted in the formation of big pharma? And I would say 90% or better will answer antibiotics. So then we now know from literature and from all the research I did, and the literature backs this up, is that chronic disease, that includes all the major diseases we have in our modern society, are caused by an imbalance from the oral cavity to the anus. So the question is, what's the primary cause of the imbalance? And then question 3, if this is causing and the imbalance, and this has been going on since its discovery after World War II, how many people have died from, let's say, the top 4, diabetes, heart disease, the neurologic disease, and most cancers are from chronic inflammation. So what single thing has caused more deaths since World War II than almost anything else? And so that answer is antibiotics. So we as dentists, we use antibiotics all the time. It is my board certification's implant dentistry, and my practice was limited to implant dentistry from the late 80s until about 5 years ago when I started phasing out of it. So spirochetes are not sensitive to antibiotic treatment. So when you treat patients with antibiotics, even one course, and the more courses, the more bacteria and microbes become extinct forever. And so that balance is disturbed, and they potentiate spirochetes. Of course, antibiotics was first discovered in 1942, so we're talking about many, many years of this, and it severely affects the microbiota. And it corresponds with this beginning of the autoimmune disease epidemic. By the way, I don't use that term autoimmune disease anymore. It's chronic inflammatory disease because it's the body's reaction to infection. There are several things that changed since World War II. And basically, when we all left Africa thousands of years ago, we basically all had the same microbes in our mouth because we all came from the same where. So over thousands of years, we spread all around the world, and we became isolated in are little small communities everywhere, and the strains over many, many years changed. So after World War II, when travel, cars, planes, and automobiles came along, we started mixing our oral bacteria together. And so it depends on robust your immune system is to the particular strain that has changed that you now have. The modern diet adds another thing, and we find that the refined carbohydrates that turn to sugar as soon as they turn in your mouth, and sugar and the whole thing is started sometime at the same time. What's the literature say? Of course, everyone in this webinar knows about Weston Price's work, so I don't have to really go over it. But he basically went to every indigenous population around the world, and he found that people not exposed to modern society didn't have cavities and they didn't have facial deformities. Sugar consumption in this country, you can see how it's dramatically increased, and spirochetes seem to thrive, and they need the more sugar, the more being out of balance already, the more populated they become. These next videos are a patient that taught me more about the immune system than any other patient, and it was just a fluke that I discovered this. But a patient came in that just had an abscessed teeth, but no periodontal disease anywhere. He came in, he wanted, because he heard about my lectures, and he said, I want to check my spirochetes. I said, well, we don't really need to. We just have an abscessed tooth. We just take it out, clean it up, and you're good to go. And he says, but I want to get it changed in. I want to have my plaque checked anyway with your microscope. So I did, and this is what I found. This was from the exudate. And those little round things we're going to talk about later, they are the spore-like forms that the body, when you have an abscessed tooth, can go in and deal with the infection, then it moves it outside the body through a draining abscess. When you look at the PMNs in these situations, I hope you can see that every one of those PMMs is loaded with microbes. And you can see spirochetes. Right there is a spirochete, and it's almost in the point where it's turning into its circular form here. And so I've never seen this before. I've been studying slides like this for years, and I never saw a microbe in any immune cell, ever. So this was a revelation to me that this is the immune system the way it's supposed to work. If you just take a plaque sample, you can have just microbes. There are spirochetes in there, the trichomonas, there are all kinds of amoeba and other things, but all of the immune cells are impotent. And what I found out later in my studies, that these particular microbes, there's spirochetes and then several others, can actually commandeer the immune system and depress it so that they can survive, and other bacteria can too. We found that over years and through the literature studies that spirochetes, chlamydia pneumonia, and a few others actually can commandeer the immune system. And we find that, and the literature backs this up, spirochetes, when you have bleeding gums and bone loss, that they can get up to 80% of the microbes that you can see. And they hijack the immune system and they produce immune suppression. Immune suppression is a common denominator among the spectrum of chronic inflammatory diseases. When we're talking about the chronic diseases, so we used to call them autoimmune diseases, it's quite an array. And if one patient has one, they usually have many, even if they're subclinical. So if they have heart disease, chances are they may have undiagnosed diabetes and an array of there's almost 100 other named chronic inflammatory diseases. They're all comorbid with each other, the comorbidities of chronic diseases. And when you study plaque, there's huge number. In fact, from the recent studies with DNA, there's twice as many microbes in the mouth than what we once saw, and most of them, half of them, cannot be cultured or characterized or named. When you get gingivitis, research has shown that 1/3 of the human genome is activated. The mouth is the primary battle for the immune system to conquer whatever it's exposed to while still in the mouth. Nowhere else in the body can you have an open wound year after year after year without getting gangrene. So if you've got an infection in your leg or something like that and it went on for more than a few days, you're going to start getting septicemia, and you may get gangrene and lose a limb, but not so in the mouth. Periodontal disease and systemic chronic inflammatory diseases. The problem is how do you diagnose them? I see a lot of papers are blaming one bacteria or another, or trying to figure it out, and it's really hard to diagnose. I mean, we're doing it with traditional probing and bleeding points, and all this stuff actually causes dangerous bacteremias. There's plenty of literature to show that. And for what we know, there's too many bacteria for DNA tests, and we don't even know what half of them are. So right now, the only way you can really appraise it is with a microscope until we get DNA tests, at least of the major players, which we don't even know what they are. I'm going to now get into why I've been writing so much about spirochetes, their morphology, the pleomorphic forms, and how they actually evade the immune system. Some of this research goes back many, many years, and when I first started understanding this, I couldn't understand how we've overlooked this. Spirochetes actually have many forms, and as they start out, the spiral forms, and then as they get older, they form little granules in anywhere from 16 to 18. And the research has shown that these little granules actually will vegetate and turn into active spiral forms. And there's another one. This is 1912. And here's one done by Alan McDonald, and he's been probably the most influential of all the researchers in this subject. And there's we call a cis form. And these are the granular forms. And here's nerve fibers, and you can see the circular forms and actually the spirochetes on nerve trunks. And he mainly was studying Lyme disease and Borrelia burgdorferi. He feels that these cis forms actually do stimulate the immune system and cause inflammation. So spirochetes get to wherever they're going to go. It's usually in this lining of blood vessels. And if you want to really get down to it, all these chronic diseases are really vascular diseases because most of the damage and where they incubate and cause inflammation is in the lining of blood vessels. These are what Lyme disease spirochetes look like. And so when you're doing your microbial analysis with a microscope, you look for the spirochetes that kind of move like snakes. Here's one right here. They move like snakes, and here's a lymphocyte that's actually engulfing one of these things. And so when you're looking at plaque, this is the Lyme spirochete, and as soon as this shows, I'm going to show two samples that I took from patients that have the same movement and the same coils as the Lyme disease spirochete in dental plaque. Here's the first patient, and as you see, most of them or some of them are moving like snakes, and that's one of the clues as to how virulent a particular spirochete is. Here's another patient. When a spirochete is engulfed within a macrophage, there's one right here, it sometimes dies, but you can see here examples of these granular forms right here. The spirochete, you can see it moving like a spirochete, and when this breaks down, these little granular forms are actually released, and when the macrophage, since in the body, it has nowhere to escape. In the mouth, it can escape through a draining fistula and be gone, but anywhere else in the body, it can't escape. So these little granular forms actually vegetate and form more spirochetes. There's a cis form up there, or spore-like form up there. This is what happens when you treat spirochetes with penicillin. This is a slide. This is a Lyme disease spirochete, and penicillin is coming from your left to the right, from bottom to top. You can see the flow of sometimes you can see the little things going by, but this is a dilute penicillin, and this is what happens to spirochetes when you treat them with an antibiotic. They react, and then they roll up into that. We call it a circular form or spore-like form, round form. There's several names for them. This is actually while it's happening. This is completely unknown to medicine. Dentists don't know anything about it. Physicians don't. Fact is, when you ask a physician what's the primary organism in the mouth that gets out of balance, they'll give you everything. Only one physician out of about 100 would give me the right answer, which is spirochetes. I'm just going to go over some of these spore-like forms where we find them, and there was one textbook back in 1960 actually described them and how they revegetate after put in a favorable medium. They'll revegetate to the regular spiral form. Here's high magnification. This is 1948 scanning micrograph. This is Wolf's work from Germany, how she actually did frozen section and showed how they morph into the circular form. Then here's actually the clinician here, the main researcher is Chang. He's got dark field and phase contrast showing these same spore-like forms. And this is Dasper. These larger, these are bacteria, they're Porphyromonas gingivalis, but the smaller ones are these small circular forms. In fluorescence spectroscopy, they show these round forms that light up the same way the spirochetes were. And there's been research done since that actually have taken these and forced them into the spore form and then put them in a medium that's favorable, and they actually turn back into the spirochete after a short amount of time. And also you find this happens to be an immune cell in the bloodstream. You find the round forms there. This is a red blood cell, and then there's one of them right there. This is blood sample, and they're free. They run free in the bloodstream as well. And it's when you're studying the blood, this is how you find the spirochetes. They're not in their regular helical form. These are the Isles of Langerhans. We're going to talk about diabetes later. And this is samples that I published where we find these in the sloughed epithelial cells that you find in the plaque samples. And when patients have severe periodontal diseases, you'll find just the epithelial cells. They're non-immune cells. The spirochetes actually invade the immune cells, and then they turn into the circular forms and then stay there until whatever treatment you've done, and then over time they reformulate into the spiral form, and then that's recurrence of the periodontal disease. The significance as far as Dasper is concerned is that's the reservoir. The gingival sulcus epithelial lining is where these things hide once you do your periodontal treatment. And as soon as the periodontal treatment's over and the antibiotics is done, a few weeks later they just revegetate and recontaminate the gingival sulcus. So one of the research that I did was on patients who had severe periodontal disease, and then I'd get samples before and after antibiotic treatment. And this is when I discovered that most of them will turn to spore form when you treat with antibiotics. And I've got hundreds of these cases where these are the spore-like forms that you find subsequent to flagyl, moxicillin or flagyl and doxycycline 2 weeks worth, 300 milligrams each for 3 weeks. And yet you'll find these spore-like forms everywhere, but no spiral forms. I got some atherosclerotic plaque from one of my patients, and we put it under microscope, and lo and behold, as I predicted I would see, in the atherosclerotic plaques, you see these spore-like forms. We're going to get into more of the meat of the lecture. We're halfway through the lecture now, and we've got to keep going here. But amyloid plaque is something that, well, amyloid, we pretty much know, or we all think we know what amyloid is, but recent studies show that amyloid is actually a normal protein, and it's an antimicrobial. McDonald, he's done a huge amount of research, and he actually has a video that if you have time to go, if I could play it for you, you can see that he shows that amyloid plaque is basically biofilms of active form of spirochetes. They have the round forms, and they're coated in amyloid plaque. In other words, the amyloid was trying to isolate the spirochetes, and it forms the amyloid plaque, which is the hallmark of Alzheimer's disease. What advantage do spirochetes have? Well, they're located in the walls of blood vessels, and they're distributed all through the body. And they spread the disease, and wherever they go is the name of whatever the disease is. And once they get into the body, they look for anaerobic conditions, sluggish blood. They have to have sluggish blood so they can actually penetrate the lining of blood vessels and set up their little camp in the lining of the blood vessel. Once they get in the body, they actually are searching for anaerobic conditions, and probably the best one is the gingival sulcus. And so that's why I see a lot of spirochetes that look like the Lyme spirochetes. So once they get into the gingival sulcus and they turn and produce spores, then the spores are transmitted through aerosol. And they get on in the food or in surfaces, they get on your hands, get back in your mouth, they vegetate, and then they've completed one of their life cycles in the mouth. And because they're so out of balance, they cause the gums to ulcerate. And then when they're manipulated, then they find their way into the bloodstream through the ulcerated gums. We know that the Lyme disease spirochete suppresses the immune system. It does so through the vitamin D receptor. And the active form of vitamin D is actually 1,25D. And the vitamin is D3. And actually, vitamin D actually is not a vitamin. It's actually a steroid. It's called a steroid. And if you look at vitamin D here, this is the active form of vitamin D. If you take this bond right here and switch it over to here, then you have cortisol. So essentially, the active form of vitamin D is very similar to cortisol. It's just almost one bond difference, and it reacts with the same receptors that cortisol. So if you have a patient that has an autoimmune disease or chronic inflammatory disease, the more severe it is, the less you should treat them with a steroid, because a steroid will depress the immune system. Well, vitamin D depresses the immune system in these patients. So it's not a good strategy to put patients, wholesale patients, on vitamin D until you know their health history and where they are on the spectrum of 1 to 100. 1 is none, and 100 is where you can't do another bypass, you can't do another stent, and the patient's basically going to die. And those patients, you can't put them on vitamin D, because the spirochetes will take the vitamin D3 and convert it to 1,25. Normally, vitamin D is produced from cholesterol from the sun, and through some sigmatic cytotrophic shifts, it goes to vitamin 2,5-D, and then it goes to 1,25-D when the body senses there's an infection. And during a normal infection, the 1,25-D is what modulates the immune system. So when 2,5-D is stimulated in the kidneys for most people, and it's also in the endometrial lining of women, which makes them more vulnerable to autoimmune diseases, because when the spirochetes suppress the immune system, then vitamin 1,25-D actually will produce more 1,25-D, which gets into the plasma, which increases in concentration. It reaches a certain concentration, and the T cells will take those and sense that the infection is over and shuts the immune system off. So basically, spirochetes produce vitamin D blockers, and then they cause the 1,25-D to increase. When that concentration increases, then the T cells sense those and shut the immune system off. You find when you have patients that have chronic disease, it doesn't matter whether it's cancer, Lyme disease, or almost all the chronic inflammatory disease, including periodontal disease, associated with decrease in vitamin 2,5-D. But most doctors or physicians will test for the 2,5-D, but they won't test for the 1,25-D. If you have low 2,5-D and high 1,25-D, you have chronic inflammation or a disease. Immune suppression links all chronic disease. So now we're going to get into when the immune system is depressed, then those patients, basically, you can find all the microbes and viruses associated with their diseases. So they basically have everything. Once the spirochetes get in and suppress the immune system, they open up the inflammatory paths, and then all of a sudden, all the bacteria can go anywhere. And so that's why there's been so much confusion as to which is causing what. Georgian Slotz, who's a friend of mine, suggested that the herpes virus in various forms is part of Alzheimer's disease. And the herpes simplex, and what you want to look at is this capsid in the middle of it, because you're going to see that again here soon. But the herpes virus number 1 is the one that seems to be associated with the amyloid plaque in Alzheimer's disease. It's found in elderly human brains. It may actually, throughout your lifetime, you are exposed to the herpes virus, and it actually becomes, it gets to the brain through what you get a breakout on the lip or wherever it is. It breaks out in the nerve fibers itself. And then when the disease is over, when the lesion heals, it migrates back through the trigeminals to the base of the brain. And I'm not sure if it's by itself can get into the brain itself. So we now know that chlamydia pneumoniae and spirochetes are found in Alzheimer's disease. They feel that the herpes gets there first, probably as a secondary infection, and probably what tips the scale that dumps these patients into chronic disease. Okay, spirochetes and Alzheimer's disease. There's really good paper that they take mice and they drill holes into the molars and they inject bacteria in there. Quite a number of different oral bacteria. But when they study these mice at 1 month, 2 months, 3 months, 4 months, at the end of that time, they find that spirochetes invade all parts of the mouse, but the other microbes don't. So spirochetes seem to be the instigator of this disease, and they can invade every part of the body. They do it through the blood vessels. So when you understand the underlying cause of all these diseases, you can begin to explain diseases. So what causes high blood pressure? And I just thought, well, it's gotten into the capillary bed, which caused damage to the blood vessels, capillaries, which increase the pressure because the blood can't get through the capillary bed as quickly, which drives up the blood pressure. So you can explain blood pressure through that. For every year you work past 65, you have a 4% less chance of getting Alzheimer's. Well, why is that so? Well, spirochetes literally have a hard time when they get to the blood. They don't really like being in the blood, so they have to do what they do fairly rapidly, and they have to find stagnant blood somewhere, so they actually have the time to drill a hole in the lining of the blood vessel. So if you think about somebody who retires, well, I've been sitting around more than I do than when I was working full time. And so my blood isn't perfusing as fast as it normally would. So I think what exercise does is that you have a vigorous pumping of blood through your system, and these spirochetes can't access the lining of blood vessels. So that's, like I say, once you understand what this is, then you can begin to understand why spirochetes are so dangerous and what you can do to evade, having those drilling holes and causing whatever disease they occur whenever. Another pathway to the brain is through the trigeminal nerve, and they transfers through the nerve, and McDonald showed that. And once they get there, they actually can penetrate the blood-brain barrier, and actually makes holes in it. So that's probably how the virus is getting into the brain. So there's 2 pathways to the brain for Alzheimer's, through the trigeminal nerve and also through the bloodstream. Another thing that I figured out, that you can make the blood more toxic to these things. And I think that's why a good diet in vegetables is so important, because when you think about it, vegetables all survive, and they survive because they have antimicrobials that are part of the vegetable. So when you eat the vegetables, then they absorbed into the system, and those antimicrobials are flowing through the bloodstream, which would inhibit spirochetes or anything else in the bloodstream. So a good diet actually prevents Alzheimer's, prevents these diseases as well. Or if you have a diet high in sugar, well, that potentiates it. So if you eliminate the sugars and refined carbohydrates, you decrease your chances of getting these into the blood, or having it actually penetrate. It actually makes the blood more toxic to these microbes, especially the spirochetes. Another thing I tell my patients is the biofilm that I showed you before, and I showed you the PMNs. It's like the PMMs form a blockade. And so the microbes can't get through this biofilm and access the blood. So water pick kind of keeps, it's like a numbers game. It keeps the microbial numbers down to the point where even if it is out of balance, they don't get a quorum enough to cause ulceration of the gingival sulcus, so that the patients they don't bleed. So when I do prophies, I've got for 30 years, because of my implant dentistry experience, we use water picks, because we found that those are basically the only thing that help implants survive year after year, because the microbes can't develop that quorum they need to cause the ulcerated gums that leads the pathway into the body. So if you're going to just do one thing for your patient, have them get on a water pick, and the microbes that are supposed to stay in the mouth will stay in the mouth. So putting it all together, I'll tell you what, since year 2000 is when the Surgeon General first came up with these papers that showed there's a relationship between periodontal disease and systemic disease. And that's when I bought my first microscope and started this research, which has taken thousands and thousands of hours since then, in both studying the microbes and microscopic examination of hundreds and hundreds of patients. But I'll tell you what, this is the most complex thing. And if I were going to say that somebody designed something for men or humans not to find, this is it. And we can get into that subject, but I'm not going to get into that subject in this lecture. But if you want to call me, which I've got my email address and my website at the bottom of each slide. If you want to email me or call me, I'm open to discussion and education. That's what I'm all about. And we can discuss in more detail as to how we put this all together. It's extremely complex and a series of seemingly unrelated steps. And I think one of the reasons this just floated between the physicians and the dentists is basically research has gotten much, much more difficult in recent years. I would say the last 20 years. As it used to be when I first started out in dentistry, you could go to the library and you could access anything you wanted to in the library. All the periodicals were up to date and you could get every article basically free. But today we have literally hundreds of associations, all have their own journals and they're not available. So if you've got a journal you can't access in normal ways, even through PubMed, you have to pay like $40 each for these articles. So doing this research has taken years and a lot of money to put these articles all together and glean what the underlying cause of these things are. And I think if Trump could do anything, he could, pay for the physicians that could have access to these articles for free. But the critical questions now is basically what is normal plaque? And what is beta amyloid? And how is beta amyloid related to chronic disease? And how is beta amyloid formed? This stuff really didn't dawn on me until very recently. And I thought I knew what beta amyloid is, but I had no idea. And I'm not even sure how I discovered it, but somehow an article clued me in and I began to understand it. Normal amyloid, and there's many articles on this, even one in the New England Journal of Medicine, that normal amyloid is actually a stacking of normal prions, and it's an antimicrobial, especially antiviral. So in its normal form, amyloid is not toxic, and it's there to fight an infection. It's pretty much primarily fighting virus. So this is something that only dawned on me or I only read about recently, and maybe, I'm not sure how long, there's an article here, 2010, but I would say in the last decade, we've been finding, the articles are pointing out that normal amyloid actually is an antimicrobial. A normal prion is to your left here. And then a diseased or folded over prion has this configuration here where the spirals are flattened out and they're folded over. So this is not normal. And so when you have a normal stacking of what we call beta amyloid, it's a stacking of folded over prions, and that's toxic to nerve cells. This Creutzfeldt-Jakob disease, CJD, that's quite personal for me because my first wife actually died from this. And before, this was several years ago. And I was told that 1 in 1 million get this. But the more research I did, the more I found out it's more common than what they purport to claim. This CJD is actually the human form of mad cow disease. And guess what? Beta amyloid is the hallmark of this disease. And researchers, over the years, felt that the prion, the folded over prion itself, called the prion only theory, one prion can cause another prion to fold over, and a chain reaction occurs that begins this process of forming beta amyloid, which is stacking of the folded over version of the prions. We now know from research that virus particles are involved. So when you think about it, when I first heard the prion only theory, I'm thinking, how can a protein cause another protein to fold over? And it just didn't make any sense to me at all. And so in my research, I found a paper that virus particles are associated with dehuman or CJD. And she feels that that gets into the DNA, the human DNA, and causes the protein or the prion protein synthesis to form folded over prions. Now that disease, or that makes more sense to me than the prion only theory. And I think that that theory will prevail. It's the minority theory right now, but I think that that theory will prevail. It's actually the DNA that's been altered by a virus particle. And so I think if we're going to look for a cure to this, is finding a way to switch that protein synthesis to normal prions, which is not toxic. Okay, plaques, this gets pretty complicated. They contain these fibroamyloid, and it's a hallmark of both Alzheimer's disease and diabetes. And when I read this, and this was the paper that I wrote for Dentistry Today, I wrote it maybe 5 months ago. I wrote it in 2 weekends and sent it to Bill Dome, and we had it ready to publish. And I sent a letter to the editor at Dentistry Today and told him, I said, I don't think there'll be another paper like this that will affect dentistry so much, because if CJD, diabetes, and Alzheimer's disease are caused by the same thing, folded over prions, which could be caused by a virus, then we got a problem, because CJD in England is a big deal. And over there, they found that patients that have CJD, there's a potential of dentists passing this virus or whatever prions between patients. And they found that the nerve tissue and root canal teeth actually contain these particular prions, and that you can't sterilize the instruments, especially the endodontic instruments, so they're prohibited from reusing or multiple use of files and our endodontic files and reamers. But because CJD is so rare, that the probability of contamination is very low. But when you get Alzheimer's disease and diabetes, now think of, and especially in my implant practice with multiple missing teeth, every single one of my patients had some form of neurologic problems, and the vast majority, if not all of them, also had diabetes. So now we have a probability of contamination between patients, and also the staff and the dentists working in that, that mist that we work in every day, the high-speed mist that our heads are right in, that is, us catching this virus and passing it along, the probability gets much, much higher. This keeps me up at night, to be honest with you, and I wake up early and thinking, how do we get rid of these? It's a daunting question, and it worries me that we are passing it around as dentists, and we have to change some of the ways we sterilize, which we don't have right now. Bill Dome, as I'm sure everybody knows who he is, he purports that ozone is the answer. But I'm not sure if it's the entire answer, because once you get ozone or get the spirochetes or whatever it is in the biofilm, it's even hard for ozone to get to it. So this is a question we're going to have to deal with as we move along in this discussion. So Alzheimer's disease, diabetes, and CJD are basically all caused by beta amyloid. Okay, now get into how these virus particles get to where they're at in the brain, as well as other places. And the research into this was very, very enlightening to me, because again, this is something I had no clue. I would say in the last year, I found this literature and started studying viruses, and started realizing that I didn't know much. In fact, as the more I get into this subject, the more humbling it becomes, because you think you got the answer, and then something comes along like this, and then realize, gosh, I don't know anything. But a bacteriophage, I don't know how many of us really knew much. Maybe you heard that term but didn't know what it was. But bacteriophages actually were used in World War II by the Eastern Bloc, Russia, to treat disease. And what it is, it's a bacteria virus, but it's very specific for the bacteria. And you can actually, through culturing, you can get a bacteriophage to target a particular bacteria. And so during the years that we were separated from Russia, they were going the bacteria route and we were going the antibiotic route. And it was very successful. Bacteriophages would cure diseases because once a bacteriophage attacks a particular virus, it injects its DNA. The DNA goes into the DNA of the bacteria, and then it produces thousands of bacteriophages, and then actually the bacteria explodes, releasing these, and then these go and find more bacteria, and it's like a chain reaction, and it wipes out the whole colony of bacteria. By the way, these are found in the ocean, very prevalent in the ocean. I think it's nature's way to control bacteria overpopulation. These things are extremely small. A bacteria, a spirochete is basically about a micron to a micron and a half in diameter, and anywhere from 5 to 30 or 40 microns long. And these things look like mosquitoes when they attack a spirochete. The problem is the research done in the bioweapons part of our government found that these spirochetes could be used as research tools. And they studied these spirochetes by injecting virus particles using bacteriophages, because bacteriophages don't kill spirochetes, but they can be used to change their DNA. And this was the research that was done, and you can do the research on Lyme disease and find that that's what they were using to study the Lyme spirochete. Spirochetes, one other thing, plastids, it's something else I didn't know a lot about. These are virus particles. They're either linear or circular. And these are associated with spirochetes and other bacteria. And that's how and these can be transported between different species of bacteria, from spirochetes to other parts and they transfer various genes from one spirochete to another to get various changes in the spirochete, and you actually can get antibiotic resistance to. That's what they did with the Lyme spirochete. But they're associated with bacteriophages, virus particles. As I mentioned before, bacteriophages' main duty is to control bacteria, and they were used in World War II. Fact is, in World War I, more people died of relapsing fever, which is another spirochete disease, than the wars, and it's like 40% fatal. And this is the spirochete that the Japanese and the Germans used spirochetes as bioweapons. And they did it through tick infestation. They'd inject them into the ticks, and the ticks would feed on the animals, and the enemy would eat the animals, and then they get relapsing fever and die by the hundreds of thousands. So the Japanese actually, there's a book called Japan's Secret Weapon, or Secret Weaponry, and then it's written in 1946, and it describes, one chapter describes the use of spirochetes as a bioweapon. They used it throughout China and down in Vietnam and through that whole part of the world. And the fact is, when I do microscopic examinations on Asians, there's not one that I've ever found that didn't have a spirochete. So we now know that the bacteriophages, the herpes virus, and the plastids are actually evolved from the same ancestral tree. So they're all basically there to change the DNA of whatever it attacks. And in spirochetes, they inject the DNA particles into the spirochete. That gets into the DNA, and they can change the virulence forms of it and actually give it antibiotic resistant and other virulent forms. So the prion theory, unfortunately, we have to deal with a sterilization problem, and we talked about the theoretical possibility of transmission, and especially now with management of Alzheimer's and diabetic patients. So we've got to work out some way, and I think there's sterilizers that use ozone, and we have to have ways of being able to test to see if it actually works. So these are the articles on ozone, and that's it. We used up our hour, and I thank you very much.
Thank you so much, Dr. Nordquist. That was a highly interesting presentation that I think gave us all a lot to think about. If anyone does have any questions before we sign off for the night, go ahead and enter them into the questions panel. And just so you know, Dr. Nordquist, Dr. Dome is on, so he was here to hear your presentation. So I know you were asking about that earlier. He says, thank you very much. So I'm not seeing any questions. Let me check again for any questions. I'm not seeing any questions coming through. So with that, well, thank you again for speaking with us tonight. And thank you, everyone, for joining us. We will see you next month for next month's webinar. Good night, everyone. Thank you.
Okay.